course, with Dr. Craig's permission, I'll go forward.
We're, I was in his last group. We're done.
You still talking to Rob or Bob? Wants to go by Bobby or Rob?
Bobby.
Bobby. This neuron should look familiar to Captain Richards.
Pardon?
I said this should be familiar to you.
This is when the images just
He also no. So after good morning. So after a conversation with a few of you, I wanted to, and this was talking about the descending pain pathway. I want to make sure we're clear of what's happening at each synapse. And in order to do that, we've got to really understand what's going on at the nerve. Right? Because just because I call something a GABAergic neuron or a glutamatergic neuron or a serotonergic neuron doesn't mean that that's the only receptors it has. The thing that means is that's what its job is to do. It's to release glutamate or to release GABA or to release serotonin. Now that GABAergic neuron or that glutamatergic neuron or that serotonergic neuron, it can be influenced by many different types of neurons. And in the exercise for the descending pain pathway I gave you, I'm just showing you some of that connectivity in the brain. Right, so if we go from that first order neuron, which is glutamate, it's releasing glutamate onto glutamate receptors on that second order neuron cell body. Right, so in your head, always remember that an axon terminal is going to release a neurotransmitter, which is then going to go and bind to a receptor on a cell body of a different neuron. So that's what this is saying. So last night I'm thinking, how can I explain this to you guys? So if you look up here, we've got this X-ergic neuron. So what's its job?
release X.
To release X. That's why it's X-ergic. It releases X. X can be whatever you want it to be. But we know that it will be inhibited or it will be stimulated by several processes, either on the cell body or at the axon terminal. Now, the different types of things that could stimulate it, for example, would be a glutamate neuron releasing glutamate onto NMDA receptors, AMPA receptors, kainate receptors located on the dendrites, the cell body of that X-ergic neuron. Or it could be stimulated by serotonin. Right? There's 7 different types of serotonin receptors with their own subtypes. Some of those are inhibitory, some of those are excitatory. In this particular example, I'm showing you an example of an excitatory stimulation from serotonin being released onto a serotonin receptor and creating an EPSP. The same goes with acetylcholine. Depending on the receptor it binds, right? If it's a nicotinic receptor in the nervous system, it's going to stimulate the neuron on the cell body that it connects to. But if it's in the nervous system and it's a muscarinic 2 in the nervous system, right? That's an example where acetylcholine could inhibit the function of that X-ergic neuron. So when you're looking at a neuron, it's named for what it does, for what it releases. So that whole flumazenil thing, that's what got me to make this. So if I were to ask about that flumazenil question, what is it doing? Is it inhibiting the actual GABA neuron? No. It's inhibiting GABA receptors on the cell body that the GABA neuron releases GABA onto. Does that make sense? There was some confusion with a few of you about that. So I want to make sure you're understanding that everything is an axon terminal synapse postsynaptic target process. Any questions on what I was trying to convey? And then what eventually will make this X-ergic neuron fire is what?
Summation at the highlight.
A summation of your excitatory influences and your inhibitory influences to see if you yielded enough electricity to send off an action potential. Right? Does that help clear up a little bit that descending pain pathway? Because that exercise is specifically done so you understand this presynaptic neurotransmitter in the synapse, postsynaptic target influence or connectivity. I mean, every nerve in the brain is innervated by just about every other type of nerve that you can think of. So if you're a dopaminergic neuron, you're having glutamate nerves coming to innervate you. You're having acetylcholine nerves coming to innervate you. You're having GABAergic nerves coming to innervate you. So we have like 10 to the 14th neurons, and each of those neurons has like 1 million connections along it. So that's how you have to think about. We show you these pictures in 2 dimensions. That's not how it happens. Everything's a summation of excitatory influences and inhibitory influences. Cool. Any questions on that? So make sure when you're going, and here's the thing for that essay. If you're talking about the activity presynaptically and postsynaptically, guess what you're doing? You're writing out the essay. If you're describing that action, that's the essay. All right, I hope that helps a little bit, because a couple of you were thinking that the actual GABA receptor with that flumazenil was